Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we discussed about the chemical digestion of food in buccal cavity, that is mouth. We discussed about the composition of saliva, and now we are going to start with digestion of food in stomach. The chemical breakdown of food in the presence of enzymes, as you know, is known as what? Chemical digestion or enzymatic digestion. And today we are going to start with the gastric digestion, that is, the digestion which takes place in the presence of gastric enzymes. So, starting with the first slide, when the food enters in the stomach, the G cells secrete gastrin hormones, which stimulate the secretion of gastric juice by gastric glands. So gastric glands, they are concerned with the secretion of gastric juice, and it is stimulated by an hormonal secretion which is given by G cells of the stomach. So G cells of the stomach, they secrete gastrin hormones which stimulate the secretion of gastric juice by gastric glands. Now, secretion of gastric juice is controlled by nerves, hormones, and chemical substances. The secretion in the presence of enzymes, hormones, the nerves, they help to control the secretion of chemical substances, that is enzymes and hormones. All is controlled by nerves, that is, we have studied about the Arabic plexus and the Meissner's plexus. Okay. So secretion of gastric juice is controlled by nerves, hormones and chemical substances. Secretion of gastric juice is divided into three phases. In stomach, the gastric juice is secreted in three phases. What are these three phases? We will learn about. First phase is the cephalic phase. This phase is mediated by parasympathetic secretions. This phase is mediated by parasympathetic secretions through parasympathetic stimulation by parasympathetic nervous system. It is the first step of secretion. It is the first step of secretion. When persons see the food, due to the optic reflex, small amount of gastric juice is secreted in the stomach due to sight. So when the person see the food, then the optic reflex gets into action and small amount of gastric juice secretes in the stomach due to sight. Gastric phase. First phase is the cephalic phase. Second is gastric phase. When food enter into stomach, then gastric phase is started. When the food enters in the stomach, then the gastric phase is started. When food particles strike to the fundic part of the stomach, then the small amount of gastric juice is secreted. When the food particles, they come in contact with the fundic part of the stomach, then small amount of gastric juice is secreted due to the reflex action and the distension of stomach. Distension of stomach means lengthening of stomach cavity. Gastric juice develops the peristaltic movement in the stomach. Gastric juice, it develops the peristaltic movement, it initiates the peristaltic movement in the stomach. Due to peristaltic, food particles are rubbed on mucosal layer of the stomach. Because of this peristalsis, the food particles, they rubbed because of the frictional force, they rubbed on the mucosal surface of the stomach, which leads to mechanical churning of food. So gastric phase, when the food enters into the stomach, the gastric phase is started when the food particles strike the fundic part of the stomach. Then a small amount of gastric juice is secreted due to the reflex action and the distension of stomach. This reflex action and the distension of the stomach leads to the lengthening of the stomach, means the gastric cavity. The gastric juice develops the peristaltic movement in the stomach, and due to peristaltic movement, the food particles, they rub on the mucosal layer of the stomach. So this mucosal layer, because of rubbing, secretes good amount of mucus, and because of this, mechanical churning starts. Okay. The stimulus of parasympathetic nervous system, that is the vagus nerve, will secrete a neurotransmitter acetylcholine. and the secretion of histamine initiates the cephalic phase. So I will write down the parasympathetic nervous systems. They are related to the secretion of a neurotransmitter acetylcholine, stimulation of vagus nerve, which is 10th cranial nerve, and the secretion of histamine. Okay, along with the secretion of histamine. Okay, so because of the secretion of histamine and the acetylcholine, which is secreted by the vagus nerve, okay, the parasympathetic nervous system gets activated. Because of this parasympathetic nervous system, the gastrin hormone, it initiates the cephalic phase or the starting phase of the chemical digestion of food. Understood? Is this clear? Now, due to rubbing process, the cells get stimulated and secretes gastrin hormones, powerfully stimulate the gastric glands for the secretion of gastric juice. Because of this rubbing process, the cells they stimulates and they secretes gastric hormone in large quantity, in large amount, which powerfully stimulate the gastric glands for the secretion of gastric juice. Some drinking substances also stimulates the secretion of gastric juice, such as soup, alcohol, caffeine, histamine. These drinking substances and gastric juice stimulates the desire of appetite. That is why these substances are called appetizers or appetizer juice. The last phase or the third phase is intestinal phase. When the food reach at the ileum, mucosal layer of ileum secretes a chemical substance. Its nature is similar to histamine or gastrin. This chemical substances goes into the stomach through the blood circulation where it stimulates the secretion of gastric juice. So first phase is cephalic phase, then the gastric phase, and then the intestinal phase. When the food reaches to the ileum, the mucosal layer of the ileum secretes a chemical substance. Its nature is similar to histamine or gastrin. This chemical substances goes into the stomach through blood circulation where it stimulates the secretion of gastric juice. Its actual cause is not known, meaning the cause of the secretion of these substances is not known yet, but it is believed that this phase starts about after 8 to 10 hours of taking a meal. So intestinal phase starts about 8 to 10 hours after taking meal. Okay? So this is about the phases of digestion in stomach, that what are the different phases of digestion in stomach. All the gastric glands are stimulated by acetylcholine secreted by parasympathetic nervous system, that is vagus nerve. I have already told you. Along with histamine also, it acts as appetizer, which further initiates the release of gastrin hormone from G cells. There is one important term I would like to make a note of it, of gastric inhibitory peptide, collectively known as enterogestrone. It inhibits the secretion of gastric juice. This hormone is released from duodenal epithelium. So please remember the gastrin hormone and anti-gastrin hormone, that is GIP, which is gastric inhibitory peptide. Gastrin inhibitory peptide act as anti-gastrin. Okay. Composition of gastric juice. The composition of gastric juice, it includes water 99.5%, HCl 0.2 to 0.3%. pH of gastric juice is 1.5 to 2.5, that is very acidic, mean highly acidic. Rest, it includes mucus, water, HCl, gastric enzymes like pepsinogen, prorenin, gastric lipase, etc. So water 99.5%, HCl 0.2 to 0.3%, pH is 1.5 to 2.5, that is highly acidic. Other than this, it includes mucus, water, HCl, gastric enzymes like pepsinogen, prorenin, gastric lipase, etc. Now, coming to functions of HCl. What are the different functions of HCl? The main function of HCl is to convert inactive enzymes, that is zymogens, into active enzymes. The main function of HCl is to convert inactive enzyme, that is zymogen, into active enzymes. HCl converts pepsinogen into pepsin. Pepsinogen is inactive form to pepsin, that is active form. Prorenin to renin. Okay. It destroys all the bacteria present in the food, means it is antibacterial. Antibacterial. HCl stops the action of saliva on food in stomach. That medium is highly acidic and salivary enzymes cannot work in such medium. So HCl stops the action of saliva in food. It dissolves the hard portions of the food and makes it soft. So it dissolves the hard portions of the food and makes it soft. So somewhat it helps in mechanical breakdown as well. Pepsinogen and prorenin are inactive enzymes. Means these enzymes, they are having basically the capping decapping process. Means the activation involves decapping. That is, suppose this is enzyme and it is a cap. This is a cap which is attached. So activation involves decapping. And the active side becomes free for the substrate. Free for the substrate. Okay. The initials of pepsinogen conversion to pepsin is done by HCl. Then pepsinogen further catalyzed its own reaction, that is autocatalysis. Means the moment initially decapping is done, which is done in the presence of HCl, then pepsinogen in the presence of pepsin gets autocatalyzed. Autocatalyzed. Okay. Digestion by renin, that is chymosin. Renin is an active enzyme which is secreted in childhood stage of mammals only. It converts milk into curd like substance, that is clot of milk, and then digests it. In adult stages, it is inactive. So renin is an inactive form of enzyme which is secreted in the childhood stage of mammals only. It converts milk into curd-like substance, that is clot of milk, and then digest it. In adult stages, it is inactive. Renin acts on a milk protein casein. Casein is a soluble protein. Casein is a soluble protein while renin is a active form of protein. So casein is a soluble protein which on acting with renin, casein in the presence of renin becomes paracasinate. Paracasein. In terms of renin, casein gets converted into insoluble form, that is calcium paracasinate. This process is termed as curdling of milk. After becoming insoluble, the milk can remain in the stomach for a longer time. For a longer time. Paracasein combines with calcium to form calcium paracasinate, that is known as what, curdling of milk. Calcium paracasinate further in the presence of pepsin converts into peptones, that are smaller peptides chains. Polypeptides. Okay. So calcium paracasinate in the presence of pepsin converts into peptones, that are smaller polypeptides. Okay. Renin is absent in humans. Clotting of milk is done by HCl in humans. In humans, renin is absent. Okay. Clotting of milk is done by HCl in humans. So renin, also known as chymosin, is an active form of enzyme secreted in the childhood stage of mammals only. It converts milk into curd-like substance, that is clot of milk, and then digests it. In adult stages, it is inactive. Renin acts on the milk protein casein. Casein in the presence of renin converts into paracasein. Paracasein in the presence of calcium to form calcium paracasinate, which is insoluble. This process is known as curdling of milk. After becoming insoluble, milk can remain in the stomach for a longer time. In humans, renin is absent. Clotting of milk is done in the presence of HCl. Digestion by pepsin. Before coming to the digestion by pepsin, I would like to tell you that when the food acts on with all these acidic enzymes and acidic pH, it is the chyme or chyme. Chyme, it includes the ingested food plus gastric juices. Chyme is basically ingested food plus gastric juice. Okay. Acid chyme moves into the small intestine. Acidic chyme moves into small intestine. No. Inactive pepsinogen on getting proper pH converts into active pepsin. Peptidase, the enzyme which breaks the peptide bond. These peptidases are of two types. Inactive pepsinogen on getting proper pH converts into active pepsin. Peptidase is the enzyme which breaks the peptide bond, and these peptides are of two types. Exopeptidase, the peptidase enzyme which breaks the outer and the marginal bond of polypeptide. Peptidase enzyme, it breaks down the outer and the marginal bond of the polypeptide called exopeptidase. In this process, amino acid and polypeptides are formed. In this process, amino acids and polypeptides are formed. Endopeptidase, the peptidase enzyme which breaks the inner peptide bond of larger peptide and forms the small peptides such as peptone, proteoses, and peptides. So endopeptidase, the peptidase enzyme which breaks the inner peptide bond of larger polypeptide and forms small polypeptides such as peptone, proteases, and peptides. Pepsin is the strongest endopeptidase. It breaks proteins into smaller molecules. Proteins and peptones plus proteoses plus peptides. So proteins, it includes peptones plus proteoses plus peptides. Okay. So exopeptidase, the peptidase enzyme which breaks the outer and the marginal bond of polypeptide called exopeptidase. In this process, amino acid and polypeptides are formed. So because of the presence of exopeptidase, amino acids are formed and polypeptides. Endopeptidase, the peptidase enzyme which breaks the inner peptide bond of larger polypeptide and forms a small polypeptides such as peptones, proteoses, and peptides. Okay, amino acids are not formed. Pepsin is the strongest endopeptidase. It breaks proteins into smaller molecules. That is, proteins, it includes proteoses, peptones, and peptides. In stomach, endopeptidase are found, so only digestion of proteins can take place properly in the stomach. So in stomach only endopeptidase is present, that is pepsin, which is a endopeptidase, which converts proteins into peptones, proteoses, and peptides. Okay. Exopeptidases, it converts all these substances into amino acids and polypeptides. Okay. Digestion by gastric lipase. It converts fat into fatty acids and glycerols. It is secreted in the less amount, so less digestion of fat takes place here. Okay. This lipase acts on the emulsified fat and convert into fatty acid and glycerol. 1% emulsified fat is present in the food. Due to the peristalsis, the food is converted into a paste. This form of food, which is thick, acidic, and semi-digested in the stomach, is known as what? Chyme. After short intervals, the pyloric valves keep on opening and closing, so the chyme is fed into intestine in installments. Okay. After short intervals, the pyloric valve or the pyloric sphincter, it opens and closes again and again, so that chyme could enter the intestine in small installments. Digestion of food in duodenum. So the digestion in the presence of gastric lipase is simple. That gastric lipase, it converts fat into fatty acids and glycerol. Okay. This lipase acts on the emulsified fat and convert it into fatty acid and glycerol. 1% emulsified fat is present in the food. Due to peristalsis, the food is converted into a paste. This form of food, which is thick, acidic, and semi-digested in the stomach, is called chyme. After short intervals, the pyloric valve keeps on opening and closing, so the chyme is fed into the intestine in small installments. Digestion of food in duodenum. When food leaves the stomach through its pyloric end and enter into the duodenum, it is called chyme, that is acidic. The HCl of chyme stimulates the wall of the duodenum to secrete hormones. It secretes various hormones like secretin. It is the most important hormone of digestive tract and is also first discovered hormone. The first discovered hormone was secretin. It is the most important hormone of digestive tract. The HCl of the chyme stimulates the wall of the duodenum to secrete hormones, among which secreting is the prima facie hormone. This hormone stimulates pancreas for synthesis and secretion of non-enzymatic pancreatic juices. Non-enzymatic part of pancreatic juice It also stimulates liver for secretion of bile juice and inhibit the gastric juice secretion in stomach and reduce rate of contraction of stomach. So basically, the secretion of secretin, it stimulates the pancreas for the secretion of non-enzymatic part of pancreatic juice. It also stimulates the liver for secretion of bile juices and inhibit the gastric juice secretion in the stomach and reduce the rate of contraction of stomach. Okay. Neutralization of acidic chyme occurs by bile juice. Okay. Pancreozymin. It stimulates the synthesis as well as secretion of pancreatic juices. Pancreozymin stimulates the synthesis as well as the secretion of pancreatic juice. Secretin promotes the secretion of non-enzymatic part of pancreatic juice, while pancreozymin promotes the secretion of enzymatic part of pancreatic juice. This is a very important point. That secretin, it stimulates the secretion of non-enzymatic part of pancreatic juice, while pancreozymin promotes the secretion of enzymatic part of pancreatic juice. Hepatocrinin, it promotes the synthesis and secretion of bile juice in liver. Hepatocrinin, it stimulates the secretion of synthesis and secretion of bile juice in liver. Cholecystokinin, it stimulates the liver and gallbladder, mainly gallbladder, to secrete bile juice. Cholecystokinin, it stimulates mainly gallbladder along with liver to secrete bile juice. Cholecystokinin. Diocrinin, it stimulates the Brunner's gland for synthesis and secretion of non-enzymatic part of intestinal juice. Diocrinin, it stimulates the Brunner's gland for the synthesis and secretion of non-enzymatic part of intestinal juice. So diocrinin, cholecystokinin, hepatocrinin, pancreozymin. Endocrinin, this hormone stimulates the Paneth cells for the synthesis and secretion of enzymatic part of intestinal juice. Duocrinin, it stimulates the Brunner's gland for the secretion and synthesis of non-enzymatic part of intestinal juice. And endocrinin, this hormone stimulates the Paneth cells for the secretion and the synthesis of enzymatic part of pancreatic intestinal juice. So duocrinin and enterocrinin for intestinal juices, cholecystokinin for gallbladder, hepatocrinin for the secretion and the synthesis of bile juice in liver, and pancreozymin and secretin for pancreas. Villikinin, it stimulates the activity of villi. Enterogastron, it inhibits the secretion of HCl in stomach. Enterogastron or gastric inhibitory peptide, it stimulates the secretion of gastric HCl secretion. Gastric inhibitory peptide, it inhibits the secretion of gastrin hormone, anti-gastrin hormone. Okay. Enterogastron, it inhibits the secretion of HCl in stomach. Vasoactive intestinal peptide VIP and somatostatin, they inhibits the motility of stomach. Okay. When controls stomach distension. Okay. So enterogastron, it inhibits the secretion of HCl in stomach. Gastric inhibitory peptide, it inhibits the secretion of gastrin hormone. Vasoactive intestinal peptide and somatostatin, they inhibits the motility of stomach, that is controls the stomach distension and all. Villikinin stimulates the activity of villi. Endocrinin, this hormone stimulates the Paneth cells for the synthesis and secretion of enzymatic part of intestinal juices. Now, we will be starting with bile juice in the next class. My dear students, till then, bye bye, take care, stay safe, stay healthy.